In this video, we will continue our discussion about C1GalT1 knockdown effect on pancreatic cancer cells. So, in this video, we will discuss how this C1GalT1 knockdown suppresses cell-ECM adhesion and what are the signaling pathways that get disrupted. Since in the previous videos we have we got to know that C1GalT1 knockdown will suppress the metastasis and migration of pancreatic cancer cells. So now they wanted to know how these suppressions of the metastasis and migrations of the pancreatic cancer cells occurs. So what they did is from the first they check this in the panel A. They check whether the C1GalT1 knockdown has any impact on the spreadness of the cells or not, and they can see that for HPAF2 and HPAC cells the spreadness goes down. The quantifications are shown in the bottoms of this panel A, where we can see that spreading cells per field is decreased in the C1GalT1 knockdown for both HPAF2 and HPAC cells. And when they check the adhere cells in both HPAF and HPAC cells, it also goes down. It indicates that C1GalT1 knockdown cells is difficult to adhere at different ECM, and they checked it for collagen 1, collagen 4, fibronectin and LAM, and they see that the adhere cells also goes down in the C1GalT1 knockdown. Thereafter, they wanted to check the signaling pathways. So integrin used to play a role for the interactions with the ECM. Integrins receptors on the cell surface can interact with the ECM for these adhesions. So in the integrins, the final molecules that get triggered is this FAK proteins. So they wanted to check whether these FAK proteins get activated or phosphorylated or not. So they use the plate that is coated with different ECM proteins and then seed their C1GalT1 knockdown cells and the control cells, and they check that in the case of C1GalT1 knockdown they see a decrease in these phosphorylated FAK signals for the collagen 1, fibronectin, and also decrease they have seen for the collagen 4 and LAM also compared to the control case, and same results they have seen for HPAF2 and HPAC cells. We can see that. And they check the phosphorylation of the at Y397 and Y925. So all this indicates that the integrins interactions with the ECM gets suppressed in the C1GalT1 knockdown cells. Now they wanted to check which domain of the integrins plays a role towards these interactions. So they did the pull down study with this alpha with different domains of the integrins, and then check these VVA lectins to get an idea about the change in the glycosylations also. And what they are able to find out is that in the case of beta 1 they see a high VVA lectins, indicates that the beta 1 glycosylations get disrupted in the C1GalT1 knockdown, which has high TN structures when the C1GalT1 has been knocked down. And they have also seen the increase for the signals for this alpha V domain and alpha 5 domain for the VVA lectins in the C1GalT1 knockdown. And these results are and for this HPAC they see an increase in signals for the alpha 5 and beta 1, and also for this alpha V, whereas not much change has been observed for alpha 2 and for alpha 2, but for alpha 2 and alpha 3. So this all indicates that the domains, this beta 1 domains plays a role where glycosylation has also been changed, beta 1 domain of the integrins, whereas the alpha 5 and alpha V also has some increase in the glycosylations or more specifically TN antigens in the C1GalT1 knockdown. So all this figure indicates that is the C1GalT1 with the C1GalT1 knockdown, the interactions between the integrin and the ECM got disrupted, and more specifically the signaling pathways, more specifically that is beta 1 domain of the integrins are expressing more TN antigens, and also there is a disruption in the glycosylation in alpha 5 and alpha V that contributes towards the suppression of cell-ECM adhesions. So I hope you understood these experimental results. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.